So what about erythropoietic porphyria? Well, I will speak only on erythropoietic protoporphyrias, mean EPP and XLP. So, as you know, these diseases result from dysfunction in other pathway enzymes, ferrochelatase and LA synthase 2. So, pathophysiology is very clear. There is a compensatory induction of erythroid 2, it means in the erythroid in the bone marrow, or there is a gain of function of LS2 in XLP. And this leads to porphyrin accumulation and acute cutaneous photosensitivity. So that's what we know. So what is actual and emerging therapy? Actually, protection from sunlight exposure is or was the cornerstone of management for protoporphyria. We all know that. And there is indeed a drug. We have an increased skin melanin production after a treatment with afamelanotide, which is a human MRC1 agonist, and it has an increased significant light tolerance, and it has an increased quality of light in a EPP patient. This has been demonstrated a long time, and the treatment was now approved by EMA and indeed by FDA, and the name of the product is Scenesse. And I think Scenesse is going to be approved by NICE in UK as well. I hope so, at least. Well, it works, but it's very expensive. It doesn't work all the time. It has some trouble indeed, and well, it's well tolerated, but it's not perfect. There will be very soon a similar treatment with a novel human MRC1 agonist, again, like Scenesse, and which is under clinical investigation, probably in UK as well, as far as I know from my colleague Bob Sarkany. The name is dersimelagon, and it's done by Mitsubishi Tanabe. So, by dealing with protection from sunlight, there are challenges for clinical trials, and we should know that. The first is how to objectively measure the extent of phototoxic reaction with the lack of specific markers to assess the clinical efficacy of therapeutics. This is a big problem, and how to identify and how to use reliable endpoints for clinical trials. And don't forget that in EPP, seasonal, meteorological, geographical variables pose enormous additional problem for those patients that always fear sunlight exposure. So that's very difficult. As a resume, it is difficult to do a clinical trial with EPP patients. So there is something new. Previously, afamelanotide Scenesse treatment was studied using time to pain or time to outside as primary outcome measure. But then, as the EPP patient have an ingrained fear of sunlight, these measures did not capture the full treatment effect most of the time. So we developed something new, the time to prodrome. It was retrospectively characterized and evaluated as a safer patient-reported outcome measure in afamelanotide-treated patients. This has been published by US and EU colleagues. And time to prodrome, just to show you, this is the feature of EPP. So indeed, you can avoid further sun exposure. You can avoid sun exposure by staying at home. But if you go outside, then you can have some prodromal symptoms, what we call warning signals: burning, tingling, itching, stinging. Well, EPP patients, they all know that they have prodromal symptoms. And if they go back at home and to avoid sun exposure, they will have no crisis. But if they continue to go under the sunlight, then they will have severe phototoxic reaction, severe pain that lasts for days, fear, behavior of sun avoidance, impair quality of life. So we use the time to prodrome to measure now the effect of the treatment, and I think it's a good way. So dersimelagon developed by Mitsubishi will use that. Dersimelagon is a novel selective melanocortin 1 receptor, as I told you. It is orally administered tablet, not injected subcutaneously like Scenesse. So you have to take that every day. Well, it's a small molecule. It increases indeed melanin density dose dependently. So what are the results for the moment? The primary study endpoint was achieved. There is a big improvement. The treatment different from placebo versus control was more than 50 minutes for 100 milligram and more than 60 minutes 300 milligram. I mean, this is the time to prodrome. Improvement of time to prodrome, which was the primary endpoint in clinical study, and the duration of sunlight exposure, which was a secondary endpoint, were observed as early as at week 6, which is very good. Treatment has an acceptable safety profile and is well tolerated. We knew that indeed with the Scenesse clinical trial before. And so now there are new therapies based on the etiologies and pathogenesis of EPP. We have first, and that's very interesting, these new therapies. We have inhibitors of erythroid LAS2. Remember, pathogenesis of EPP is a compensatory increase in LAS2. So if we have inhibitor of that, we can probably treat EPP or lower PP9 production. We can also modulate PP9 distribution because we know that we have a transporter called ABCG2, which modulates the efflux of PP9. And so by inhibition of this, we can modulate the distribution of PP9 probably in the skin. And so the third is partial suppression of erythroid heme synthesis. If you have an increase in heme synthesis in the bone marrow producing a lot of PP9, you can try to lower LAS2 activity by suppressing one of the substrates, and glycine is a substrate. And so we have now a new glycine transporter inhibitor, which is called bitopertin, and is developed now by Disc Medicine. And we have also, but I will not speak about that because too much complicated for today. We can also try to suppress the hypomorphic fake allele, the famous IVS3 minus 48TC polymorphism, which is found in more than 95% of EPP. If you suppress the effect of this hypomorphic allele, maybe you can try to treat patients, but it's very difficult to reach the precursor cells in the bone marrow. So I will not speak about that because it is for the moment, it's not continuously developed. So what's bring Agios? Agios is a pharmaceutical company in Boston, and they have developed an inhibitor. And they have developed first, they have generated an XLP mouse model with an LAS2 mutation by using the famous CRISPR-Cas9 mechanism. They have achieved in vivo proof of concept of treating erythropoietic by targeting LAS2 by SHRN knockdown. And then they have developed, they have determined, that 50% LAS2 protein suppression was sufficient for normalization of blood PP9 in XLP mice. And they have demonstrated that they have a small molecule, an LAS2 inhibitor, that can lower bone marrow LA and PP9 level on acute dosing. We have no news since that because this slide was given to us in 2017 in Bordeaux ICPP meeting. And we have no news since that time. I think they have trouble with the small molecule. It's probably as a side effect. I don't know exactly. But this is something that could be developed. They're still working on that. So what about the transporter ABCG2 I speak about? So we know that because of the work, it's developed by Bridge Bio, a company in the West Coast in the US. And the paper was published by Wong et al. And we know that ABCG2 deficiency decrease PP9 distribution to the skin, and therefore prevents EPP-associated phototoxicity. We know also now that ABCG2 deficiency protects against EPP-associated hepatotoxicity, which is quite nice because the risk of liver cancer is low, but it exists in EPP by modulating PP9 distribution, metabolism, and expression. So the essential role of ABCG2 in the pathophysiology of EPP suggests strongly that there are novel strategies in the development of therapy for EPP. And so for the moment, BridgeBio tried to develop a molecule that inhibits ABCG2. So probably we will have news about that. For the moment, I have no news, but they are working on a drug which can inhibit ABCG2 and that modulate the distribution and lower the concentration of PP9 under the skin, and so lowering the intolerance to the light of EPP patient. So, and now we have a new drug. Well, at least before that, you know that PP9 is a key point of therapeutic intervention in EPP. Indeed, if we can lower PP9, we can cure the disease. So, how we know that? We know that from this slide, you know. You have different patients. There are patients with lower PP9 level, and they have an increased light tolerance in EPP. So you can see here, low PP9, they can tolerate much more the light. And we can say that 25% of patients with low PP9 experience symptoms versus 75 to 100% experience symptoms where they have medium or high PP9 level. So it means that even if you don't suppress completely PP9, that if you can lower PP9, that you will have a good effect and you will lower the experience of symptoms. And we know also that because we were able to do interventional extracorporeal photoinactivation of PP9. This has been done once at least. And if you have from that experiment, we know that 30% reduction led to an average 14-fold increase in daylight tolerance. So it means if you reduce PP9 by 30%, you have a much better tolerance to the light. And another experience, which is your experience, is that women, EPP patients during pregnancy, women with EPP during pregnancy, they experience decrease in PP9 level, and they may lead to temporary reduction in symptoms. They can lie to the sun, you know. I've seen many EPP women like that during their pregnancy. It starts after the second trimester of pregnancy. It's very funny, and the plasma PP9 level is really low in the blood, probably due to the fact that the baby may probably play a role in lowering PP9 in the body. So we have a new drug that does that. It's called bitopertin. Bitopertin is a drug that was developed by Roche in Switzerland, and it was done to try to treat schizophrenia. But they have done a phase 3 study, and at least it didn't work. So, but you know, this drug lower LAS2 activity because it reduces the uptake of glycine in developing erythrocyte by lowering LAS2 activity. And so that's very interesting. And so Porphyria people from Switzerland and after that from in the US by Disc Medicine, a company in the West Coast as well, they have developed this drug to try to treat EPP patients. So if you go to EPP and XLP patients, so you have an increase in LAS2. Indeed, you have a defect in fact, in EPP, an increase in LAS2 in XLP, but an increase in LAS2 in both. And so these mutations result in a reservoir supraphysiologic levels of PP9. We know that, okay? So if you treat by bitopertin, you can reduce and normalize PP9 level because you reduce the glycine uptake, so LAS2 activity is reduced, and so you reduce the production of protoporphyrin 9. So it can cure possibly EPP and XLP patient. And if we look at the cellular model, which is KR562 EPP cell model, and so you can see that bitopertin can inhibit PP9 accumulation. This is already percentage of inhibition. You can see it's a huge inhibition, and the production of heme is almost unaffected. So we will have more data coming soon from animal model, but I think it is, to me, it is very promising. It's very promising because bitopertin has been shown to have absolutely no adverse event during the clinical trial in human for schizophrenia. Indeed, it's schizophrenia, it didn't work, but this is not the problem. The problem is the drug was safe. So, it will facilitate a clinical trial of bitopertin in human. And I think this medicine that has now the property of bitopertin, they will develop very soon a clinical trial in human using bitopertin. And maybe we will have there a good treatment. Indeed, I'm not sure that bitopertin will lower completely PP9 production, but don't forget that if you are able to lower by 30 to 50 or to 60% PP9, you will have much less light intolerance in EPP patients. So I'm open for question indeed, and thank you for your attention.